Hey guys, welcome back to my channel. This is Dr. Rajeshwar from YR PharmaTube. In the previous video, we have learnt the reduction reactions of drug metabolism. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and other subjects of pharmaceutical sciences, click on the links given in the description box below this video. In this lesson, we shall try to understand the hydrolysis reactions of phase 1 drug metabolism. At the end of this video, you will be able to understand the process of hydrolysis and the different types of phase 1 hydrolysis reactions, which include hydrolysis of esters, hydrolysis of amides, hydrolysis of hydroxides and carbamates, and miscellaneous hydrolysis reactions. Before going to understand the hydrolysis reactions of drug metabolism, let's take some keynotes of hydrolysis and its definition in chemistry. Hydrolysis is any chemical reaction in which a molecule of water breaks one or more chemical bonds. The term is used broadly for substitution, elimination, and solvation reactions in which water is the nucleophile. Hydrolysis reactions can be the reverse of a condensation reaction in which two molecules join into a large one and eject a water molecule. Thus, hydrolysis adds water to break down, whereas condensation builds up by removing water. Therefore, in reaction with water, a bond in the compound is broken, resulting in two compounds. At the same time, the water molecule splits into two with a hydrogen transferring to one of the compounds and a hydroxide to the other compound. See the reactions. In drug metabolism, hydrolysis is an important major metabolic reaction for drugs containing groups, namely esters, amides, nitriles, ethers, hydroxides, and carbamates. Ethers in biosystems undergo an oxidative cleavage, that is O-dealkylation, which was already discussed in the previous lessons. Nitriles or cyanides are divided into two categories, simple alkyl and aromatic. Simple alkyl cyanides may result in cleavage to inorganic cyanide ion. They exhibit the toxicity characteristic of inorganic cyanide, that is metabolized to thiocyanate by the mitochondrial enzyme rhodonase utilizing thiosulfate or mercaptopyruvic acid as the sulfur donor. Aromatic nitriles appear to be totally resistant to enzymatic hydrolysis. The aromatic nucleus is instead oxidized in the manner as previously described in the aromatic hydroxylation reaction. Thus, the hydrolytic reactions of biological interest as far as the drugs are concerned are primarily those of esters and amides. Some enzymes that perform hydrolysis reactions include esterases, peptidases, and amidases. Hydrolysis reactions generally involve acid derivatives. The products of these hydrolysis reactions are generally more water soluble than the original compound and are excreted from the human system. The four major types of hydrolysis reactions include number 1, hydrolysis of esters, number 2, hydrolysis of amides, number 3, hydroxide and carbamate hydrolysis, and number 4, miscellaneous hydrolysis reactions. Number 1, hydrolysis of esters. Hydrolysis is a major biotransformation pathway for drugs containing an ester functionality. This is because of the relative ease of hydrolyzing the ester linkage. The general scheme for hydrolysis of esters is illustrated in the figure. This type of reaction can take place in the plasma, liver, kidney, and other tissues. In plasma, non-specific enzymes such as acetylcholine esterases, pseudocholine esterases, and other esterases catalyze the reaction. And in the liver, specific esterases catalyze the hydrolysis for a particular group of drugs. In these reactions, esterase enzymes in the plasma and liver inactivate or detoxify a relatively nonpolar molecule by splitting it into two polar or water-soluble components such as an alcohol and an acid. One of the best examples of ester hydrolysis is the metabolic conversion of aspirin, an analgesic agent, to salicylic acid. The ester group present in the salicylic acid can be cleaved by the hydrolysis to form two products, namely salicylic acid and acetic acid. In the other example, procaine, which is an anesthetic agent, can also undergo hydrolysis, and this reaction is catalyzed by a plasma esterase enzyme to form the corresponding alcohol and an acid. In the third example, pethidine or meperidine is hydrolyzed by the liver esterase to form its corresponding acid and alcohol metabolites. In the fourth example, of the two ester functional groups present in cocaine, which is an anesthetic agent, it appears that, in general, the methyl group is hydrolyzed preferentially to yield benzoyl agonine as the major urinary metabolite. The hydrolysis of cocaine to methyl agonine, however, also occurs in plasma and to a minor extent in blood. Methylphenidate is hydrolyzed rapidly to give ritalinic acid as the major metabolite. In a majority of ester drugs, ester hydrolysis leads to pharmacologically active metabolites. For example, hydrolysis of diphenoxylate, an antidiarrheal agent in humans, leads to diphenoxylic acid, which is about 5 times more potent antidiarrheal agent than the parent ester. The presence of esterases in many tissues and plasma makes ester derivatives logical prodrug candidates because hydrolysis causes the ester prodrug to revert to the parent compound. The rapid metabolism of clofebrate yields para-chlorophenoxyisobutyric acid as the major plasma metabolite in humans. Studies in rats indicate that the free acid, the product from the reaction, is responsible for clofebrate's hypolipidemic effect. Many parent drugs have been chemically modified or derivatized to generate the so-called prodrugs to overcome some undesirable property such as bitter taste, poor absorption, poor solubility, irritation at the site of injection, etc. The rationale behind the prodrug concept was to develop an agent that, once inside the biological system, would be biotransformed to the active parent drug. The presence of esterases in many tissues and plasma makes ester derivatives logical prodrug candidates because hydrolysis would cause the ester prodrug to revert to the parent compound. Accordingly, antibiotics such as chloramphenicol and clindamycin have been derivatized as their palmitate esters to minimize their bitter taste and to improve their palatability in pediatric dosage forms. Number 2. Hydrolysis of amides. Generally, amides are resistant to hydrolysis in water even after prolonged heating. However, in the presence of an acid or base, hydrolysis proceeds at a moderate rate. The general scheme for amide hydrolysis is illustrated in the figure given here. Thus, hydrolysis of an amide gives a carboxylic acid and the salt of ammonia or an amine. In living cells, amide hydrolysis is catalyzed by several enzymes present in the liver called the amidases. Amide hydrolysis is also catalyzed by some non-specific esterases as well as by non-specific amidases, carboxypeptidases, aminopeptidases, and decyclizases. These enzymes are found in various tissues in the human body. As mentioned above, most amides are hydrolyzed by the liver amidases. Theoretically, amides may also be hydrolyzed by plasma esterases, but such reactions proceed more slowly. For example, the deacylated metabolite of indomethacin, which is a tertiary amide, has been detected in the human urine as one of the major metabolites of indomethacin. Some of the metabolites are detectable in plasma, and the free and conjugated metabolites are eliminated in the urine, bile, and feces. The occurrence of enterohepatic cycling of the conjugates and probably of indomethacin itself is an important feature of the metabolism of this particular drug. Between 10 and 20% of the drug is excreted unchanged in the urine, in part by tubular secretion. On the other hand, it is important to emphasize that in certain cases, the reaction of hydrolysis may liberate the active metabolite of a parent drug. A case in point is that of thionyl sulfathiazole, which under the action of bacterial enzymes in the colon liberates the antibacterial agent sulfathiazole. Ethyl glycyl xylidine, the N-deethylated phase 1 product of lignocaine, is hydrolyzed by the liver microsomal fraction to yield xylidine and ethyl glycine. Similarly, penicillin undergoes hydrolysis to its corresponding acid metabolites. More specific enzyme systems are able to hydrolyze sulfate and glucuronate conjugates as well as hydrate epoxides, glycosides, and other moieties. In all these various reactions, the products formed may be subsequently converted by phase 2 reactions into inactive and or more water-soluble conjugates that are more easily excreted. It is to note that the hydrolysis of esters is usually rapid, while that of amides is often much slower. This makes esters suitable as prodrugs and amides a potential source for slow release drugs. As discussed above, amides are hydrolyzed slowly in comparison to esters. Consequently, hydrolysis of the amide bond of procainamide is relatively slow compared with hydrolysis of the ester linkage in procaine. Drugs in which amide cleavage has been reported to occur include lidocaine, carbamazepine, indomethacin, and prazosin. Amide linkages present in barbiturates, for example hexobarbital, as well as in hydantoins, for example 5-phenylhydantoin, and succinimides like fensuximide are also susceptible to hydrolysis. Number 3. Hydrazide and carbamate hydrolysis. Less common functional groups present in drugs such as hydrazide and carbamate groups can also be hydrolyzed. A hydrazide can be hydrolyzed to form a carboxylic acid and hydrazine, while carbamates form carboxylic acid, which is a carbonic acid, and a secondary amine in the form of dimethylamine. A well-known example is the hydrolysis of the hydrazide group of isoniazide, an anti-leprotic drug, forms isonicotinic acid and hydrazine. In this context, the hydrolysis of proteins and peptides by enzymes can also be mentioned here, but these enzymes are mainly found in the gut secretions and are involved only in a small extent in drug metabolism. Exceptions occur in the further metabolism of glutathione conjugates and in the metabolism of orally administered peptide or protein drugs. And number 4, miscellaneous hydrolysis reactions. Hydrolysis of recombinant human peptide drugs and hormones at the nitrogen or carbon terminal amino acids by carboxypeptidase and aminopeptidases and proteases in blood and other tissues is a well-organized hydrolytic reaction. Examples of peptides or protein hormones undergoing hydrolysis include human insulin, growth hormone, parathyroid hormone, and atrial natriuretic factor. In addition to hydrolysis of esters and amides, hydrolytic cleavage of other moieties occur to a minor extent in drug metabolism, including the hydrolysis of phosphate esters, for example, diethylstilbestrol diphosphate, sulfonyl ureas, cardiac glycosides, carbamate esters, and organophosphate compounds. Glucuronide and sulfate conjugates also can undergo hydrolytic cleavage by beta-glucuronidase and sulfatase enzymes. Also, the hydration or hydrolytic cleavage of epoxides and arene oxides by epoxide hydrase is considered a hydrolytic reaction. This is the list of references followed for the lesson. That's all in this video, the phase 1 hydrolysis reactions of drug metabolism. In the next lesson, we will discuss about the hydration and miscellaneous phase 1 reactions of drug metabolism. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.